Okay, we're going to go ahead and get started here. So good morning, good afternoon, good evening, good night to everyone, depending on where you are signing in from. It's my pleasure to kick off Emcee Sensory and Motor Systems and Physiology with our first keynote here at Neuromatch 3.0. And today we are going to have a talk from Marge Livingstone from Harvard University. And the title of the talk will be Category Bias Domains in Inferotemporal Cortex. How do we get them? Go ahead and take it away.
All right, you've got my slides, right?
Yes.
All right. We know from neurology and functional imaging that humans have specialized domains in ventral temporal cortex dedicated to processing faces, places, text, bodies, or tools. These domains could be innate, given that we have domains for biologically important categories. And most importantly, these domains are all in stereotyped locations. But experience must play a role in domain formation, given that we have domains for unnatural things like buildings and text. We obviously have a lot of early experience with text and with faces. To look at the role of experience in domain formation, we taught young monkeys to recognize and discriminate a set of human symbols. When we scanned them with fMRI, we found that they developed domains selectively responsive to the trained symbols. This means that experience is sufficient for domain formation. I guess we already knew that from the fact that humans have text domains. There are a lot of theories out there as to how domains form, that they're purely innate for recognizing biologically important things or similarity to biologically important things like tree branches and text, what we use them for, like text areas being preferentially connected to language areas, when during development some category is intensively experienced, and lastly, how expert you have to be to discriminate within some category. So to try to disentangle the effects of expertise, age of learning, and functionality, we taught a bunch of monkeys to recognize 3 distinct sets of symbols. And we taught these different symbol sets in different orders in different monkeys. And to our surprise, training order didn't matter, level of expertise didn't matter. Each symbol set developed a domain in a stereotyped location. So these fixed locations indicate that indicate some kind of experience-dependent sorting of inputs on some kind of a proto-map. We looked at the early development of face domains in baby monkeys. Earliest in development, we didn't see any face domains. By about 200 days of age, we could reliably detect face selective activity, and this selectivity was stable in these regions after that. The fact that domains are absent at birth, the stereotyped locations of domains, and the fact that experience can cause the formation of unnatural domains implies some kind of proto-organization, again, that experience acts on to cause the formation of these domains. But another possibility is that domains just need to mature. So to distinguish between these two ideas, we asked what happens if you block a particular experience. We raised baby monkeys while we wore welder's masks and didn't let them see any other monkeys. In control monkeys, you have face domains, and these are the face domains again. Face-deprived monkeys had no face domains. So it could be their entire temporal lobe was messed up by the abnormal rearing conditions. So we showed them images of things that they saw a lot of, like hands, human hands, monkey hands, and familiar objects. So controlled monkeys have face domains, and adjacent to that, they have hand domains. Face-deprived monkeys have no face domains, but they have hand domains, and if anything, stronger hand domains than control animals. And they had normal scene and body domains. So experience must be necessary for domain formation. The stereotype locations, the requirement for experience, again says to us their experience must be acting on some kind of proto-architecture. So to figure out what this proto-architecture was, Michael Arcaro looked at the data we had collected from newborns and analyzed the patterns of spontaneous activity across visual cortex. So in adult monkeys, retinotopic maps of visual space are found throughout the visual system, even in regions that textbooks refer to as non-retinotopic. And eccentricity, so this is eccentricity, is mapped throughout the visual system in adults. Michael found that functional connectivity patterns in these newborns define a large-scale network virtually identical to the architecture of the adult visual system. And by seeding chunks of V1 with known retinotopy, known eccentricity, he found a large-scale map of eccentricity in newborn monkeys, virtually identical to the large-scale eccentricity organization we could measure in the same monkeys when they got older. So you must start with a map. Experience sculpts this map into functional domains. But how do you get the stereotype locations? Well, there are two features of category domains that might be involved. Face domains are localized to central visual field parts of IT and scene domains to peripheral parts. And face domains respond better to round things like balls and clocks, and scene domains to rectilinear things. So how are these three things related? If category is the fundamental organizing feature of IT, maybe face domains become centrally biased because we look at faces, and scene domains become peripherally biased because we view scenes peripherally. Maybe face domains respond to curvy things because faces have a lot of curvy contours, so curvy things look more face-like than trees, and anything straight looks more like it could be part of a scene than a face. We mapped eccentricity and curvature in our monkeys. We confirmed that face selective domains, so here's a face selective domain, prefer curvy things and prefer central visual field stimuli. But when we looked at the entire visual system, all the way back here, there was a huge correlation between curvature and central visual field and rectilinearity and peripheral visual field. We were surprised by this because nobody had mentioned it before, and we discovered that a lot of previous studies just mask off V1. The correlation between eccentricity and curvature, because it covers the entire visual system starting in V1, is therefore more fundamental than the correlation between either one and category. Xiaomin Yue and Leslie Ungerleider's lab recently found the same thing is true in humans. These are 4 maps from 4 different humans. Red is curvy and blue is rectilinear, and curvy maps to central visual field and rectilinearity to peripheral visual field. Again, all the way back to V1, covering pretty much the entire visual system. Acuity varies dramatically with eccentricity, and receptive field size varies with eccentricity. So central visual field has teeny weeny receptive fields, and peripheral visual field has huge receptive fields. David Hubel and Torsten Wiesel found in both cats and monkeys that there were a lot of cells that were selective not only to orientation, but also contour length. So this cell responds strongly to a short bar, but not at all to a long bar. Such end-stop cells like to respond to corners. David and I found a lot of end-stop cells in V2, maybe more even than in V1. So this cell responds to short bars and not long bars. We found lots of them and they were clustered. We suggested that they would be particularly responsive to curved contours as well as corners. And the degree of curvature that end-stop cells respond to will scale with eccentricity, with central visual field responding to tight curvature and peripheral visual field to straight contours. The fact that these 2 studies both found such a strong correlation between curvature and eccentricity means that end stopping must be pretty prevalent. But I don't think we should think of it as just end stopping or just curvature or just retinotopy, because at every stage of the visual hierarchy, inputs are combined to generate more complex receptive field properties. And response properties of so-called neurons in an artificial neural net show how converging inputs might combine to make complicated feature selectivity, which must happen at every scale must happen at different scales for every eccentricity. And such a combinatorial mechanism might result in something like Finley Bao and Doris Tsau's lab reported recently. They found that there was a gradient of shape selectivity in monkey IT with selectivity for faces, which are blue, on the lip of the STS, which maps to central visual field, and then grading to spiky things as you go more peripherally. So it's not just curvature, it's some combination of curves and eccentricity and so on. And if you look closely at the shapes characterized by this gradient, there are a lot of round contours in the face and stubby domains and a lot of straight lines sticking out in the spiky domain. So a retinotopic map automatically gives you a low-level shape map, which could bias that part of IT to be selected to faces, which are made up of a lot of curvy contours. But could where on the map you tend to look at things also bias the development of categories? So Peter Shade asked, what do monkeys look at? And here's a video of a normal monkey looking at contours in my lab coat and my face, a lot at my face. That there is the face of God. And here's a face-deprived monkey looking a lot at my hands, contours. So if you look at the looking pattern of control and face-deprived monkeys, everybody likes to look at contours and everybody likes to look at small, round, dark things. Control monkeys look mostly at my face, but face-deprived monkeys like to look at hands and faces, but mostly hands. So face-deprived monkeys spent more time looking at hands than at faces, and control monkeys spent more time looking at faces than at hands. So control monkeys have face domains and control monkeys look at faces. Deprived monkeys lack face domains and they do not look at faces. Do domains cause looking behavior, or does looking behavior contribute to domain formation? Control monkeys looked more at faces, which are the filled symbols, than at hands long before face domains developed. So face looking behavior precedes domain formation. Is face looking innate? So even early in development, face-deprived monkeys look more at hands than at faces. So face looking can't be innate. Yet there is a huge literature showing that newborn humans and newborn monkeys track faces. This is usually done by seeing whether the infant tracks a moving schematic face. But there's some recent evidence that what infants track are small dark things, especially in the upper visual field. And the part of our brain that makes us track small moving dark things is our evolutionarily old superior colliculus. And this paper by Hafford and Chen reports that there is an upper field bias in monkey superior colliculus. And I suspect that an upper field bias is evolutionarily very old because a lot of land animals have upward facing eyes because that's where trouble comes from. So I suspect early infant face looking may be driven by the superior colliculus, not face domains in IT cortex. You start with a map. You start with a map, experience acts on that map biased by a map-based shape gradient and by looking behavior. What I'm proposing is activity-dependent clustering of inputs, the same mechanism that's thought to generate the stripes and domains in V1 and other primary sensory cortices. At birth, The inputs to V1 are segregated into ocular dominance domains, which are thought to form because prenatal waves of activity in the two retinas are independent and therefore uncorrelated. And the activity-dependent learning rule is that correlated inputs are reinforced and uncorrelated inputs are weakened because the correlated inputs succeed in firing off the postsynaptic cell. At birth, V1 is organized into orientation pinwheels, again thought to be generated by activity-dependent sorting of on and off inputs and lateral connectivity that encourages neurons to retain inputs similar to their neighbors. People used to argue about maybe they still do, what ocular dominance columns and orientation columns are for, what their function is. But Peggy Law and Martha Constantine Patton implanted a third eye on tadpoles, and the tadpoles grew up into three-eyed frogs. And when they injected the third eye with a tracer, they saw ocular dominance columns in an animal that normally never has ocular dominance columns because normally frogs don't have two eyes projecting to the same tectum. That means that ocular dominance and orientation columns aren't for anything. They just emerge as a consequence of a Hebbian clustering rule. So can we possibly attribute the large domains in IT to the same mechanisms that form the much smaller domains in V1? Shaheen Nasser and Roger Tutel's lab mapped ocular dominance columns in humans in V1 and color and disparity columns in extrastriate cortex. The domains in V2 are coarser than the domains in V1. And there are domains in V3 that are still coarser than the domains in V2, and maybe even domains in V4 that are even coarser still. So maybe domains just get larger going up the visual hierarchy, possibly due to input convergence at each stage and therefore broader correlation patterns. If so, huge domains in IT wouldn't be all that surprising. So I'm proposing that these self-organizing mechanisms acting on a map are universal. How universal? Well, at least down to frogs and beyond just vision. So here's the map of visual eccentricity in a newborn monkey. And here's a body map encompassing both somatosensory and motor cortices in a baby monkey. You can even see finger maps, which indicate that the body map is actually multiple mirror image maps of the body. And if you combine the visual and the body maps, plus the fact that this bit right here is multiple auditory tonotopic maps, then more than half of the newborn monkey brain consists of maps of the sensory periphery. So that's pretty universal. So let's say you start with a map, which is set up by trophic gradients of trophic molecules, and then prenatally that map is refined and clustered by spontaneous activity. Postnatally, the same neurons get driven by stimuli in the environment, and the same rules now form functional clusters based on what the animal experiences in its environment, including faces. So here's a recording from a face neuron and 2,550 images rank ordered by how one face cell responded. So all the top images are faces, and if I blow up the top part, you can see that they're all faces. Here's another cell in the same face patch, in the same monkey, and its top ranking images are people wearing PPE. Personal protective equipment. That's what we wear when we go into a monkey room. There is no way a monkey could have evolved selectivity for people wearing PPE. That must reflect this animal's individual experience. Now, our image set included things the monkeys were familiar with, so you might worry about image bias, but there's the same kind of bias in any natural image set. So Wiltzow developed this program that uses a neural network and a genetic algorithm to evolve optimal stimuli for a recorded neuron using image codes, which means a big image space, then selecting codes based on the neuron's response, mutating, recombining them over and over to maximize a neuron's firing rate. So we start with random textures, and then over a few hours, the synthetic images gradually evolve to maximally stimulate this neuron. This neuron on one day evolved this image. And then that's the image it evolved one day, and then the next day and the next day. So 3 independent evolutions for this same neuron with a chronic recording electrode. And its favorite natural images were monkey torsos. Everybody in the lab, when they looked at these evolutions, said, "That's Anthony." Anthony is the monkey who lives across the room from the monkey we were recording from. Note the red collar. Here are evolved synthetic images and best natural images for 4 neurons, and the evolved images do look like some kind of archetype or face template that you could have evolved. But there are other neurons that evolve things that were clearly specific to that animal's particular experience. So this is what this neuron evolved over a few hours on one day. And everybody who in the lab who saw this said, these are the rank order images for that neuron. Everybody said, "That's Diane," who's the person who comes around and gives treats to all the monkeys every day. Again, there's no way this animal evolved selectivity to a human face wearing a blue thing around their head, unless that's what they saw every day. I recorded from this cell many times, and it always evolved something rectilinear and grayish. And this cell's favorite image was always the food hopper in the monkey room. So all these selectivities show evidence that experience sculpted neuronal selectivity. In the last few years, though, there have been a number of fMRI studies on blind human subjects who were presented with tactile or auditory stimuli that resulted in category selective activations localized to similar parts of ventral temporal cortex as in sighted subjects. So these 2 studies show that tactile responses to faces or auditory responses to face-like sounds like whistling or kissing in blind subjects localize in or near what are face-selective visual areas in sighted subjects. So the interpretation of virtually all these studies is that there must be some innate predisposition of this region to become category selective, no matter what modality provides the input. It's not just visual, and that this bias is driven by top-down influences from some higher area in the cortex. In this study, a social area is proposed to provide top-down input that causes the face area to become selective for faces. These studies show that blind subjects reading Braille, tactile, or auditory input associated with text activate a region that in sighted subjects is the visual word form area. So again, the interpretation is that some higher areas feed back to temporal cortex to cause selectivity for faces or for text. So let's incorporate both these 2 summary diagrams into the simpler one. So now I'm not taking somebody else's stuff, I'm fiddling with it. So the proposal is that language areas selectively feed back to drive text selectivity in the visual word form area, and social areas feedback to drive face responsiveness in the face area. This study reports that sounds associated with either faces, objects, body parts, or scenes activate the same gradient of categories as images do in sighted subjects. So let's add scenes, objects, and body parts to requiring top-down influences on the specification of temporal cortex. This study reports that the medial-lateral gradation in the temporal lobe from animacy to inanimacy categories found in sighted subjects is also found in blind subjects to auditory input. So let's add animate and inanimate gradation to the top-down requirement. And these studies find that small manipulable things and large immovable things presented auditorily to blind subjects map to similar locations as they do for images in sighted subjects. So this much higher level feedback specifying the organization of ventral temporal cortex. I have trouble with this idea. Not the results, just the interpretation. First, because higher areas evolved later than early areas, so this idea just seems backward to me. Also, recent studies from Lena Ibrahim and Gordon Fischel's lab show that although there are indeed a lot of feedback connections in adults, during development, there are many more feed-forward inputs than feedback. Also, feedback inputs are required for the, feed-forward inputs are required for the development of the feedback inputs. So this isn't a mouse, but it's also true in monkeys. During development, feed-forward inputs precede the development of feedback inputs. So I want to simplify that diagram on the grounds that Bria Long in Talia Kunkel's lab found that both the big-small gradient, big-small, and the animate-inanimate gradient in sighted subjects could be accounted for by low-level shape selectivity, because you saw the same thing when you use these scrambled up images whose identity is unclear but still retains the low-level shape selectivity. So let's simplify it to needing to explain just text, scenes, objects, body parts, and faces now. I think we can explain the localization of text, face, and scene areas for visual input just on map-based shape selectivity and how you tend to look at things. But how can we explain the cross-modal auditory and tactile activations in blind subjects? I want to invoke a global organization of maps in cortex. In somatosensory and motor cortex, upwards on the body is ventral in cortex, and down in the body is dorsal. It's also true in visual cortex, and indeed the same trophic factors, efferents, set up the gradients for the different sensory modality maps. And if you look at visual and body maps on the monkey brain, although there are multiple maps and sometimes the maps go around corners, largely down in the world is dorsal in the cortex and up in the world is ventral. Let's consider scene processing. Parietal cortex has a scene area and is generally involved in navigation. It is adjacent to peripheral parts of the retinotopic map. And in parietal cortex, there are somatosensory visual neurons with congruent visual and somatosensory fields. Also, some parietal cells have auditory input with corresponding auditory localization fields. So I think we can explain at least some multimodal scene mapping in blind subjects by congruence between the different sensory maps. What about faces? That's a little harder. Connectivity within inferotemporal cortex is generally along isoeccentricities. Central representations tend to interconnect with central and peripheral with peripheral. So you just go along isoeccentricity. This connectivity is antero-posterior. If we continue that connectivity, central visual field aligns with low frequency in the auditory map, which is where vocalization or speech is localized, and eventually with the face part of the body map. So I speculate that social areas in frontal lobes are social because they get convergent, congruent inputs from face IT and from vocalization areas, not the other way around. And that connectivity from the visual word form area ends up projecting to language areas because it's used for language and those connections are selected by use, not that language areas start out connected to what will become the reading area. I'd even like to come up with a feed-forward explanation for language areas, but now's not the time for that. So far from finding these studies on blind subjects challenging, I think they may be revealing universal principles of how different sensory modalities are aligned and interconnected. And I think these principles are universal among mammals. Different sensory maps are aligned according to the parts of the world they represent and interconnected so that, unlike us, a lot of mammals don't have to learn to integrate visual and motor or somatosensory or auditory information. They're born able to do that. And I think we inherited that alignment and interconnectivity, and that's what's being revealed by these blind subject studies. Given that we evolved from animals who don't have higher cognitive areas, it seems to me that we ought to be looking for bottom-up explanations rather than top-down. So to summarize, there are no face patches in face-deprived monkeys. Experience is necessary for domain formation. We have symbol patches in symbol-trained monkeys. It's sufficient for domain formation. So experience is necessary and sufficient for domain formation and doesn't require any innate predisposition. So we propose that experience acting on a protomap could drive the formation and localization of modules using the same kinds of universal activity-dependent clustering mechanisms that lead to orientation and ocular dominance in early sensory areas. Thank you. And I am happy to take questions.
Okay, well, thank you so much for such a fascinating talk. And as someone who does not study vision, I really appreciated how you walked through the material at a level that even I can understand. So thank you for that. So for the Q&A session, we have a couple of options here. There are some questions that have been submitted through the question and answer function here in Zoom. So we can, either I can pose the question, or we can, if you would like to ask your question yourself via video, we're also able to do that. So let's see. So we have the first question here. Okay, so let me see. For our back end help, I'm not exactly sure how to send a message to people who are asking questions. Okay, so Ben Dean has the first question, and he said he doesn't need to use his video. I'll go ahead and present it. Okay, so he said your behavioral results, which are the lack of face preference in face-deprived animals, disagree with the name, I believe, is Yoichi Sugita's 2008 results. Is there an explanation for this discrepancy?
Yes. If you look carefully at Sugita's results, he states that, so the result that Ben is talking about is that he face deprived a bunch of monkeys and then asked whether they preferred to look at faces versus objects. And then once, if they experienced it, did they prefer to look at faces versus objects? And it turns out, although they did, the face deprived monkeys looked a little bit more at faces than objects, it turns out that difference was not statistically significant. So our results do not disagree with Sugita's results. What he finds is that after experience, they look more at whatever faces they saw, but not right after the deprivation. That result's not significant.
I just have a follow-up, a clarification there. I thought and maybe misinterpreted the figure that you showed results that the face-deprived monkey, or comparing the face-deprived monkeys with the controls and their preference for faces versus rectilinear?
Hands.
Hands, okay, okay. So there's a bias toward hands in the face-deprived monkeys, but that's still true, okay.
But they don't look, Sugita looked at faces versus objects. And he didn't get a statistically significant difference in the face-deprived monkeys.
Great.
And I actually, because it wasn't very clear, and he says one thing in the text and another thing in the statistics, I actually emailed him and asked him, and he said, right, that's not significant. Because yes, it is confusing.
Great. Okay, the next question is from, let's see, Hans, I'm gonna Optebeck. And would you like to ask your question via video? Feel free to respond in the chat. Okay. Given the timing, I'm just going to go ahead and pose the question. Oh, did I hear him?
I don't know how you.
Yeah, he said no to the video.
Okay, thank you. So he said a lot of great ideas from your talk. And why does the explanation have to be bottom up, visual, or top down? these do not have to be mutually exclusive, I think.
Oh, well, first of all, I spent so many decades in V1 that I really do tend to be a pretty hardcore bottom upper, but evolutionarily and developmentally, bottom up is all you need. And it seems to me that if you're going to invoke a top down explanation, that's more complicated. And I think you need stronger evidence than the bottom uppers need.
Okay.
I think there's some name for that.
The simpler explanation?
Yeah. Occam's razor.
Yeah, the more parsimonious explanation, I guess,
Yeah.
is maybe the term. Seems like only you and I are talking here.
It's just a matter of, I think it's a matter of taste, really, whether you like bottom up or top down, but I think the bottom up is the simpler explanation.
Great. Okay. The next question is from Lena Ali Ibrahim. Do the face domains span the entire cortical column?
I'm not sure anyone knows. You'd have to go into the face domain perpendicular to the surface, and I don't know that anybody's really made a thorough study of that. I certainly don't know.
Okay. Let's see. Next, we have Dana, I believe, Bobinger. Very interesting talk. Thank you. You make the claim that VWFA becomes connected to language regions because it is used for language. But how does this explain results from Zeynep Sajin's work that show that the localization of VWFA can be predicted in individual children before they can read based on the connectivity profile of that region?
Because I think that the connectivity profile may be connecting low-level maps, earlier maps, rather than something special having to do with language. Again, I prefer the idea that they're bottom-up commonalities that we inherited from other mammals, rather than something human-specific that we have to invoke.
Okay.
You skipped the one about face-deprived monkeys have behavioral deficits.
Oh, I don't know how I did that. So, oh, and you can see the question. So go ahead.
I don't know. We tried testing them for face recognition, but it turns out monkeys will do whatever they have to do to get juice, and they learn to discriminate faces. But it was hard to. I'm pretty sure that it was. Our test was limited. I think they would pick out some particular feature on whatever face we were getting them to try to match and just go with that. So they did learn to discriminate faces, but I think they were cheating. I mean, they may have been fine at it, but monkeys usually cheat on tests like that. So I don't know the answer.
Okay. All right. So we have another question here from Lena Ali Ibrahim. The question was, is there a critical period for face domain formation?
Gosh, I wish I knew. Do you have any idea how expensive these experiments are? We'd have to do a series of them, and we're talking a year of raising a many $1,000 animal. So it's just not going to happen.
I had a question regarding the experiments that I really found fascinating, the experiments you showed where you essentially use, I believe it was maybe a genetic algorithm to establish the features that were most selective for given neurons.
Well, we call them super stimuli. We evolved
super stimuli. And so would you then expect that different super stimuli, so within the same animal, the same region, if you looked across different neurons, that they would be responsive to different super stimuli, similar? Yes. Okay.
But we never, so we use chronic microelectrode arrays, and they're spaced right now far enough apart that we don't know about nearest neighbor neurons. What we know is that neurons that are, a few hundred microns apart tend to, one gets really good, the other one doesn't particularly care about it, and so on. Lots of times if you're in a face patch and you optimize, so those archetypal faces I showed you, they tended to set off the whole population.
Okay. Okay. I have a couple more questions here. So this one, I wish I had institutions here as well so people could find each other easily. So you see, so the question is from Cesar Vargas. How contextual or stable do these image selectivities tend to be? In other words, if you did the image mutation experiments to determine selectivity in a different room in the building, would that neuron still exhibit the selectivity determined previously? So contextual learning, I like these questions.
I like that too. That's a very, you must work on the hippocampus. I love it. The selectivities can sometimes be weird in that, so there are two answers to that. There are some neurons that like both faces and hands, and sometimes the evolution takes off towards the hands and sometimes towards the faces. So I think the optimum stimulus isn't one thing, but it's like a bunch of things that are probably functionally related. And the second answer is Mike Arcaro and I have a paper that, I forget where it came out. eLife maybe? On the fact that face neurons get facilitated by bodies below the face. So if you put something in the face neurons receptive field that's not a face, but you put a body under it, it gets more activated by that non-face thing. So I think they are contextual, but not in the place cell kind of a way that you're talking about. So does that answer that question?
Let's see. Yes, he said, really like the result. He works in motor cortex, but thinks about context often. Okay, so next we have a question from, just disappeared, Nancy Conwisher. Beautiful talk, but I don't totally get your account of how the lateral fusiform becomes face selective, but not curvature selective in congenitally blind people.
Not curvature selective. So I think if you look at that old paper about parietal lobe conjunction between somatosensory and visual, it's more like what part of the world they have in common rather than low-level shape parameters that they have in common. So I don't think that if you looked at the neurons that are particularly responsive to central visual field and mouth, that they're necessarily going to share all those features, but what they will share is face and central visual field. So I think it's common maps. I think it's interconnected maps, not up and down.
All right. Okay, we have a few more, we have a couple minutes. So I guess the next question in line is from Isaac Moran. Does cortical neural code change depending if the animals are trained to recognize faces in a task in comparison with natural recognition? So
first of all this plasticity has to happen in very young animals, right? And so you can't, I think you can manipulate the selectivity in inferotemporal cortex in adult animals by training them, but really big changes, I think you need to start with a young animal, in which case you're going to have to modify their early visual experience. And I am still writing a grant right now to do exactly what you're suggesting. Great. But training is not the right word for it. It's got to be like rearing conditions, I think.
So because of the format of this, I think we may get kicked off in seconds. So I just want to thank you, Marge, for the talk. I want to thank everyone who attended. It looks like we had somewhere between 80 and 100 participants, which I think is really great for this type of format. Hope everyone stays engaged throughout the week. This is Neuromatch, so try to find people, connect, get to learn about different work that is being done all over the world here. And with that, we'll wrap up and end before we are punted away. All right. Thank you, everyone.
Thank
Take
you.
care.